Oh, here's a good one. The protozoans of the nervous system. I'll tell you again. This one we're talking about right now will be on your lab practicum. So please know this. African trypanosomiasis, also called African sleeping sickness. There's the pathogen is Trypanosoma brucei, protozoan, infects humans, and it has two subspecies. So when it gets into your bloodstream, then that protozoan invades the brain tissue and causes a coma-like condition. That's why they call it African sleeping sickness. So the subspecies are Trypanosoma brucei gambiense, that's West African trypanosomiasis. Humans are the main reservoir. And after you're infected, there's very few symptoms for weeks or months. When you start getting symptoms, you get a fever, headache, get deterioration of the central nervous system. And if you don't get any treatment, it can lead to coma and death. The second one is Trypanosoma brucei rhodiense. It's East African trypanosomiasis. And this one infects humans and domestic animals. Symptoms are within a few days, and death is within a few weeks or months. Transmission of this protozoa is by the bite of the tsetse fly. The tsetse fly is a vector that is taking that protozoa and giving it to somebody else. So the tsetse fly becomes infected when it bites an infected host that can be an animal or a human, and then that protozoa gets into the fly, and it's kind of like with malaria and the mosquito. The trypanosome multiplies in the fly's gut, and then it goes to the fly's saliva, and it develops into the infectious stage in the salivary gland. So the next time then that the fly bites you, then it's going to release a fully formed stage of the parasite into the wound. The trypanosome moves into the lymphatics and the blood. It has a virulence factor, and that is when the host starts producing IgM antibodies against that protozoa, the trypanosome. Then that trypanosome changes its surface antigens, making existing IgM ineffective, because your body makes antibody, which is IgM in this case, against a certain antigen. And the antigen is on the surface of the trypanosome. And so when that happens, then that trypanosome changes those antigens. So that antibody is no longer effective against that antigen. So then your body makes new IgM antibodies to the new antigen. And then that trypanosome changes its antigens again. That's called an antigenic shift. And this can go on, but eventually the host immune system is going to get overwhelmed and is not going to be able to produce effective antibodies. Diagnosis is by microscopic exam of blood and cerebrospinal fluid, and prevention is vector control. Yes? Oh, one thing that they do is that they make, don't ask me how they do this, but they sterilize male flies and then they release them out into the environment so that they can mate with the female. So she's gonna lay her eggs, but they're not going to be fertile. But I don't know how you round up a bunch of male flies to sterilize them. Treatment, fluornithine, developed in 1992. It's able to cross the blood-brain barrier, and it blocks an enzyme that is required for the trypanosome to proliferate. And it's effective against Trypanosoma gambiense even in the late stages of the disease. This is a slide that will be on your lab practicum. And you should just get familiar with this picture. Download it, put it someplace. Know that that's gonna be on there. And you looking at that slide will be able to tell me the name of the protozoa and the disease that it causes. That's the tsetse fly. Glossina morsitans, also known as the death dealer. Somebody donated their arm so it could bite them.
Oh, nice.
Amoebic meningoencephalitis, caused by 2 species of protozoa. Primary amoebic meningoencephalitis is caused by the amoeba, the pathogen, Naegleria fowleri. It's a problem worldwide, and we have a few cases in the United States. Transmitted by swimming in ponds and streams. And usually it's children that are gonna get this. Because the amoeba is down in that, if you've ever been to a lake, that silty part that's the bottom of the lake, that's where the amoeba live. And children go in and they start stirring up the water and splashing. Yeah, and that's how they get it. Initially, it infects the nasal mucosa, so you get this water in your nose with the amoeba in it. And then later it's gonna penetrate the brain and proliferate, but it gets to your brain through the olfactory cranial nerve. So it's gets into the nasal mucosa, gets to your nerve and travels up that nerve and goes to the brain. Fatality rate is almost 100%, and that's within days after symptoms appear. Destruction of the brain and spinal tissue causes hemorrhages and coma. And the problem is diagnosing this. If they could diagnose it right away, they might be able to do anything, but it's so quick after they get it, and they don't, the doctors, of course, don't think to check for this. There was a boy in California out with his family in a lake, and he was dead within a week. They didn't, and the diagnosis was made after he died. Excuse me. That's a picture of the amoeba, Naegleria fowleri, becoming more of a problem. Amoebic, also causing amoebic meningoencephalitis is caused by a pathogen called acanthamoeba. It causes granulomatosis amoebic encephalitis. Transmission is by direct contact. And the portal of entry isn't really known, but they think possibly the mucous membrane. It is a chronic, slowly progressive disease and can be fatal in weeks or months. And this amoeba causes multiple lesions in the brain, granulomas that form around wherever the amoeba is. So there's a picture of Naegleria, the amoeba. Prions. Prions are those abnormally folded proteins. They affect the nervous system. They have a long incubation period, maybe years. They damage the central nervous system. It's progressive. You get spongiform degeneration of the brain. So transmission is direct contact with infected tissue. And diagnosis is biopsy or image of the brain, imaging of the brain. There's no treatment. Prions are heat and chemical resistant, so you can't kill them by autoclaving or anything like that. So one of these prion-caused diseases is what's called sheep scrapie. Affects sheep. Causes the animal to scrape itself against fences and walls until it's raw. The animal gradually loses motor control and dies. This prion can be passed to another animal experimentally by injection or infected brain tissue. Of infected brain tissue. So they know that it can go from animal to animal that way, or that's why they don't want animal feed for animals that don't usually eat meat, like cows. That's a picture of a sheep with sheep scrapie, and you can see where he rubbed off his wool off of his bottom. Using his wool. In humans, the disease is Creutzfeldt-Jakob disease, or CJD. It's rare. There's about 200 cases a year in the U.S. It often occurs in families, and so they think it's a genetic component. But families are together, it might be something environmental too, in my book anyway. It can be transmitted by corneal transplant, injection of growth hormone. Those are ways it's been spread from infected human tissue. And boiling or radiation or autoclaving are not, you can't destroy that prion by those methods. In New Guinea, the tribe suffered from spongiform encephalopathy, and they called that disease Kuru, and it used to be transmitted to these tribes through the practice of cannibalism. They used to kill their rival and then eat their brain, and this prion is something that attacks your brain. And hopefully there's no more cannibals.